Hello, my name is Eduardo Noel. This is the video entry graded version for week 5 biochemical testing for microbial identification, carbohydrates, fermentation testing for Bio 225. Today is June 9, 2020. Here is my card. On my last video, I showed the results of my experiment, and I will repeat it again. This is the glucose epidermatitis, glucose cerevisiae. There's both of them. Fructose. This one right here is the epidermatitis, and cerevisiae right there. Gas fermentation and acid production. And this is the mannitol epidermatitis right here, and cerevisiae of mannitol right here, orange in color, while the rest of the fructose and glucose showed yellow in color. I believe that my experiments were very successful. There was a color change. Nothing were still red. So off on the glucose and fructose turned yellow within 12 hours of incubation, and the mannitol turned into red, orangey, fermented during the 12-hour incubation. So there was positive results on my experiments for this lab week 5. How does this apply to life is basically simple sugar. Simple sugar with the bacteria, epidermatitis and cerevisiae are the life source of those bacteria to able to live, because the color change that we know that the bacteria is still alive, and the production that it caused. Basically, on the glucose and mannitol showed that that there was gas production and acid production with the bacteria that was still alive and been refrigerated for weeks and out through end. I was amazed how those two bacteria could still produce as long since I created them on week 1, and they're still alive, and things that they could easily do with the current simple sugar that it has to survive. Mannitol is also a type of sugar alcohol, but which contains 60% fewer calories, and is half sweeter than compared to the glucose and fructose, but it showed color of orange. Didn't show that much gas production. I was looking through a Durham tube. I think what I saw was stains of not gas, but liquid trying to pour in. But when I put the Durham tube earlier, it was empty. So I think it was just stain of liquid. I don't think there was any gas production on the mannitols. But the results were orangey, and there was positive results on color change. It didn't stay yellow. So that's a plus, and the plus is that all my experiments showed results. With this experiment, on one of my videos, I had to stop the video to confirm the Durham tube because I was a bit lost, and I was looking at one of the reference pictures that indicated that we have to put the tube inside. And I was like, how? I had a test tube. I was thinking to myself, like, how does a test tube fit in such a small hole? So I had to re-look at my paperwork, the items of list that was provided, and make sure that I had everything I needed. And I saw Durham tubes in there compared to the testing tubes. So try to look for the Durham tubes through the mess that I have, and I finally found it, and it worked. And I was amazed that I was like, that was impossible for that test tube to fit in through the current broth base for the pheno. So I was kind of glad that, I avoided that problem or wasted time. So I just took the time, slow down, and read it carefully. So that's the only error I made on this experiment. Was just basically I need to double check my materials, especially I was a bit confused with the test tube and the Durham tube, and I was looking at the pictures. That was impossible. So I was able to figure it out after a bit of finding out and researching it. So for this video, that's it for this video for week 5. Thank you so much.